Hey everyone, Dr. D here, and in this video I am going to explain cellular respiration as well as fermentation. And this is going to be a one-of-a-kind video. This is my way of explaining this topic. I know it's super complicated and convoluted, and it could be one of the harder things to learn in a biology course. So I'm going to do my very best to make it as straightforward as possible, and I'm going to explain it in my own unique way that makes sense of it in a story-like fashion. So you're learning the story of the process so you can make better sense of it. So let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
So what is cellular respiration? This is how our mitochondria produce ATP. And it's not just in eukaryotic mitochondria like this one, but prokaryotes as well can undergo cellular respiration. You ever heard in grade school, the mitochondria is the powerhouse of the cell, that cheesy saying? Okay, and why do they say that? They say that because the mitochondria produces ATP, and the mitochondria produces that ATP by using chemical energy that you've consumed in your food, right? So you ate sugar like this. See, this is glucose, C6H12O6. This is glucose. I'm gonna write that down. And usually when we're discussing cellular respiration, we're talking about how glucose is broken down by the mitochondrion to form ATP. But just realize that, fats are also broken down via cellular respiration. Even proteins can be broken down via cellular respiration. But we're going to look at how glucose, we can use the energy from the glucose molecule to make ATP for the cell. Remember, ATP is the energy currency of the cell. Anytime the cell wants to do an endergonic process, a process that requires energy, then the cell can utilize ATP to drive that reaction forward, right? So the cell constantly needs ATP. Your body constantly needs ATP. And the reason you eat breakfast, lunch, and dinner, the reason why you consume sugars and fats and proteins, one big reason is to get chemical energy, right? The chemical energy that's going to produce that ATP. And that process, this equation right here, this is the equation of cellular respiration. This is cellular respiration. I'll write that right here. Cellular respiration. Okay, so cellular respiration. Let's go over the equation real fast, and then I'm going to show you exactly how cellular respiration works. So take a look here. Glucose and 6 oxygen are required. These are the reactants. These form 6 CO2. This is a product. This is CO2, carbon dioxide product, and 6 water. This is also a product. Okay, they form 6 CO2, 6 water, and either 30 net ATP in eukaryotes or 32 net ATP for prokaryotes. I'll explain why later on. But so from one glucose molecule, we are able to get, in us eukaryotes, we can get 30 ATP molecules, right? And then our body can use that ATP to do things that need energy to work in the cell. Okay, but the story all starts in the mitochondrion. So take a look here. This is a mitochondrion. And what I need you to remember before I start my story are the parts of the mitochondrion, right? So remember this. Let's just do a review of the cells chapter. Remember cells chapter? We have an outer membrane. This is the outer membrane of the mitochondria. See what I drew here on the right? This is an animal cell. Remember, animal cells have a nucleus, and then they have various mitochondria inside. They can have dozens of mitochondria. This is one mitochondrion. That's why it ends in on, right? But you would have numerous mitochondria. And I'm just showing you a mitochondrion has an outer membrane. And what's this membrane? Do you remember what this membrane is right here? That's right, Wicket. It is the inner membrane, Okay, and let me ask you this. What's this space in the middle called? What's that fluid in the middle called? Do you remember? That's called the matrix. Remember that? The matrix. And this one's tough. What was the space called? You see that space between the outer membrane and the inner membrane? That tiny bit of space between the outer membrane and the inner membrane of the mitochondrion? Yes, this little space is called the intermembrane space, okay? I just need you to remember that because I'm going to show you what we're going to do next, okay? Bear with me. I know it seems convoluted. It's going to be a long story, but I promise you if you follow me step by step, you will understand this. I want you to understand this. And I want you to not just memorize things, but I want you to really get it, okay? So we're going to do that together. What I'm going to draw next is I'm going to zoom in, okay? I'm going to zoom in on this part of the membrane. I'm going to zoom in on that part of the membrane, and I'm going to magically make this part of the membrane pop up on that board. There you go. So I have now zoomed in. What I'm trying to show you, let's get our bearings real quick. What I want you to understand is that I'm zooming in on this little box right here, looking at these two membranes of the mitochondria, right? Do you remember what was this membrane called, the bigger membrane? That's right, Wicket. The outer membrane. So this is the outer membrane. This right here is a phospholipid bilayer. It's the outer membrane of the mitochondria, okay? So this here is, I'm just going to write OM for outer membrane of the mitochondria, which would make this membrane the inner membrane. I'll write IM for inner membrane of the mitochondria. Again, what was the tiny bit of space called between the two membranes? You got it, Wicket. The intermembrane space. This is your intermembrane space. Remember, this is this tiny bit of space between the outer membrane and the inner membrane of the mitochondria. All right, now let's think about this. If this is the outer membrane, that's the inner membrane, what's this space represent? If we were to zoom in on this box right here into the mitochondria, this area right here would represent what part? Can you beat Wicket? Hopefully you beat Wicket, but Wicket is right. This is the matrix. This is the matrix of the mitochondria. So this is the matrix of the mitochondria. This represents the inner membrane of the mitochondria. This space represents the intermembrane space of the mitochondria. This membrane represents the outer membrane of the mitochondria. And then what would be this out here? What do you think that is? If you said cytoplasm, this is the cytoplasm of the cell, right? Outside of the outer membrane is the cytoplasm of the cell. So this represents the cytoplasm of the cell, okay, of the eukaryotic cell. Okay, so hopefully we have our bearings at this moment. Now I'm going to be doing some erasing, so if you're following along, if you're trying to draw this out, I recommend that you use pencil and eraser or color pencils that can be erased, because I'll be doing some erasing as I explain what's happening in the mitochondria, like how the mitochondria is making ATP. So let me introduce you to a very special enzyme. Okay, it's a membrane enzyme complex, and do you know where it lives? It lives in the inner membrane of the mitochondria. See, I'm going to do a little erasing here, like I said, and I'm going to draw a special membrane enzyme. This guy right here, I'm drawing him in purple. Okay, there you go. Now, what is this thing I drew in the inner membrane of the mitochondria? This is an amazing enzyme. It's an amazing machine, and I want to explain it to you because it will blow your mind. This enzyme is a transmembrane. Remember, transmembrane means it spans the entire membrane. It's a transmembrane enzyme, and the enzymatic activity that it does is that it can take ADP, remember what ADP stands for? Adenosine diphosphate. And it can also take a PI. Do you guys remember what PI stands for? Inorganic phosphate. And guess what it can do with the two? This enzyme can actually combine the two. It actually combines ADP with P, or inorganic phosphate. What do you think it makes when it combines ADP and P? That's right, Wicket. It makes ATP. Okay, it makes ATP. No exclamation mark necessary, but it makes ATP. And this is an amazing enzyme that makes ATP. And actually, do you remember how many ATP are made during cellular respiration from a single glucose? What can you tell me? From a single glucose, we can make either 30 net ATP for eukaryotes or 32 net ATP in prokaryotes. Well, guess what? 28 of those 30 or 32 are made right here by this special enzyme. So I'm going to name this enzyme for you now. This enzyme is called ATP synthase. Synthase. Do you know what that stands for? Remember, enzymes end in ase. So this is the ATP synthesizing enzyme, ATP synthase. And its job is to live as a transmembrane protein. And you can find them in the inner membrane of the mitochondria. See this? The folded cristae of the inner membrane of the mitochondria is littered with these ATP synthases. There isn't just one. There's the thousands upon thousands of these ATP synthase in the inner membrane of the mitochondria. In fact, the inner membrane of the mitochondria is highly folded because you have to have tons of space for as many of these ATP synthase as possible, right? There'll be tons and tons of them. And what are they all doing? They're all making ATP. And how do they do it? How do they make ATP? They combine ADP and P to make ATP. Isn't that cool? So this is why it's called ATP synthase. Now, let me ask Wicket. Wicket, what are you curious about this enzyme? Let me ask you, Wicket, and let me ask you at home. Do you at home think that ATP synthase is just making ATP spontaneously? Remember what spontaneous meant? It meant energetically for free. It meant without any energy input, right? Let's see. You got it, Wicket. Making ATP is not free, is it? Breaking ATP, if you break ATP into ADP and P, that releases a bunch of energy, right? That's spontaneous. So do you think the opposite, making ATP, do you think what ATP synthase is doing, making 28 ATP for every glucose molecule, that it's energetically favorable, that it's spontaneous, and it's just doing this for free? No, right? So what I'm trying to get at is that something must be powering ATP synthase, right? Something must be making it work. Okay, and here's something I'll tell you about ATP synthase. It's so cool. Listen to this. You see the top part I drew here? The top part of ATP synthase literally spins, okay? Everyone, look at this. This top part literally spins. If you look at ATP synthase, it has a top portion that can rotate. And it looks kind of something like this, okay, from your textbook. It looks something like this. And this top portion right here, it can rotate, okay? And look, the bottom portion here it can produce ATP. All right. And so let me ask you this. Do you think this top portion can just spontaneously spin and pop out ATP? No, right? No. Something is driving the top portion to spin. And yes, this is a rotary motor. Isn't that neat? It's the world's smallest motor. The top part literally spins so that the bottom part can make ATP. Isn't that neat? So if the top part doesn't spin, the bottom part doesn't make ATP. So something must be driving the top part to spin. Do you get where I'm going with this? Something is causing the top part to spin. And I usually ask my students to answer, what do you think is driving the top part to spin? I'll give you a second to come up with an idea. What do you think is driving the top part to spin and providing the energy so that the bottom part can make ATP? No, you're actually wrong. You guys, this is the first time Wicket has ever been wrong. He must have been slacking this week. Wicket, it's not ATP. It's actually something else. Isn't that neat? ATP is not what's driving ATP synthase to spin. Actually, something else is. Let me share with you what. Let me share with you. Well, it turns out that out here are a bunch of protons. Do you guys remember what protons are? Let me do an aside real quick. Do you remember what a proton is? A proton is H plus. Do you remember that? H plus means proton. H plus means proton. Hydrogen ions are protons. And that means that a hydrogen is simply a proton with an associated electron. Remember that? This is important to remember. There's a reason I'm reminding you about this. Electron. So a hydrogen atom is a proton with an electron. A hydrogen ion is simply the proton. The electron went away, okay? Well, it turns out there's a bunch of protons out here in the intermembrane space. Look at this. I'm going to draw a bunch of H pluses in this little space. Look. There are a bunch of H plus. In fact, what I'm going to do is this. Watch, I'm going to draw brackets around one of these H plus, and I'm going to put an arrow up showing you there's a high concentration of protons, okay? There's a high concentration of protons in the intermembrane space. And the matrix, remember this is the matrix down here, the matrix has protons as well, but the concentration of protons is lower. So there are protons here, but there aren't nearly as many, right? It's not as concentrated, I should say. So low concentration of protons on this side of the membrane in the matrix, high concentration of protons on this side of the membrane in the intermembrane space. So now let me ask you this. Do these protons, if they're highly concentrated in this little space, do these protons want to diffuse into the matrix where there's a lower concentration of protons? I'll give you a second to think about that. You have totally redeemed yourself, Wicket. Yes, the protons want to diffuse back in, right? But let me ask you this. If you've been paying attention over the last few chapters, if you've been paying attention, let me ask you this. Can protons, which have a charge, can they diffuse directly through a membrane to the other side via simple diffusion? No, they can't, right? Because they're charged, right? Charged entities, charged substances like protons, they can't diffuse across a membrane. But that's where ATP synthase comes in, you guys. Look at this. ATP synthase, the reason the top part spins is because it allows protons to stick. The protons will stick to the top part, and then the protons actually force their way in, and they actually cause the spinning, and then the protons force their way into the matrix. Isn't that neat? So just like the Hoover Dam, you know, if you've ever heard about the Hoover Dam, there's water that flows from the top part, you know, the higher level of water through the dam to the lower part of the water, the lower water level, and that drives turbines that spin. Okay, this is based on not gravity, right, but on diffusion. The protons are forcing their way in because there's a high concentration of protons in the intermembrane space. The protons force their way into the matrix. They can't go through the membrane directly, but they can go through ATP synthase. You know why? Because ATP synthase serves as a carrier protein. Remember carrier transporters? ATP synthase will allow the protons to get to the other side, but at a cost, right? The cost is spinning the ATP synthase. Kind of like, you know, if you're at the airport and they have those rotary doors, you know, those revolving doors. How does the revolving door work? Let's say it's not an automatic one. Well, you have to come up to the revolving door and you have to push on that revolving door, right? And that causes the revolving door to turn, right? And the revolving door turns, but it's because you're pushing on the revolving door, right? Does that make sense? Well, this rotary part of the ATP synthase, it turns, but the only reason it turns is because the protons are pushing, like people pushing out of the airport, turning that door, right? So as long as the protons are diffusing in, the top part spins. It has power. It's powered to spin. And that's what's providing the energy to make ATP. Remember energy coupling? I said that an exergonic process can release the energy required for an endergonic process. Well, here, the exergonic process is the diffusion of protons. And the endergonic process that it's powering is the formation of ATP, 28 ATP. And this is known as ATP synthesis. And this is why this enzyme is known as ATP synthase. Now, a little bit of terminology for you guys. I know I'm beating you up. I've gotten a lot of information across, but believe me, there's a lot more to cover in cellular respiration. But if you're ever confused, hit stop, go back, listen to it again, draw it out. You can do it. You know, this is going to be tough the first time. You may have to listen to it a couple times, but I promise you, you'll get there. Now, I said diffusion of protons across a membrane, right? Diffusion of protons. Did you know that has a name associated with it? That name is, I'm going to write it here for you. Diffusion of protons, it's termed chemiosmosis. Chemiosmosis is the diffusion of protons. That's what's powering ATP synthase to spin and to produce ATP. Okay. Now here's another terminology I need you to know. Okay. When the protons undergo chemiosmosis, don't they exert a force? Like as the protons are diffusing in, aren't they producing a force that's causing this rotary part of ATP synthase to spin, right? Well, that force has a name, okay? I know it's getting tricky. That force, the force that's causing ATP synthase to spin is called PMF. PMF for proton motive force, okay? So think about it this way. Chemiosmosis, diffusion of protons across a membrane, exerts a force. That force is called the proton motive force, or PMF for short. That's the energy that's being provided to spin ATP synthase so that it's powered up to produce ATP. Let me put it this way. If those protons weren't forcing their way in with chemiosmosis, exerting a proton motive force, then ATP synthase, the top wouldn't be spinning, and the bottom would not be producing, right? It would not be making any ATP, okay? So now, let's turn our attention to the next thing. I hope everyone's up to speed. Again, let's just recap. ATP synthase, an important transmembrane protein complex enzyme that exists in the inner membrane of the mitochondria. Actually, there's thousands of these guys in the inner membrane of the mitochondria, in those cristae, right? Now, the top part of ATP synthase spins to power the bottom part to make ATP, okay? And the reason the top part was spinning, remember, was because of proton flow, right? Protons diffusing across the membrane, process known as chemiosmosis, exerting a proton motive force. Now, let me ask Wicket. Wicket, what do you think, if we're trying to understand this process, what question pops into your head as the next logical thing to ask about how this process works? What do you think, Wicket? What are you guys curious about at home? Like what is the next thing that you'd want to know in order to understand this process? Let's see what Wicket says. Okay, that's great, Wicket. Wicket's asking, well, how is there so many protons out here? Do you see this, you guys? There's a high concentration of protons in this intermembrane space. You see this? And there's a lower concentration of protons on this side. If this was a closed system, do you remember what would happen? All these protons would flow in, or at least half of these protons would flow in until there's an equal concentration of protons on both sides. And let me ask you this. If all the protons squeezed in and there was an equal concentration of protons on both sides of this membrane, would ATP synthase still be spinning? That's right. That's right, Wicket. No, it would not spin. So here's what I'm getting at, you guys. Something must be keeping the proton concentration high in the intermembrane space. It's not like you were born, right? It's not like you were born with an infinite amount of protons in the intermembrane space, and they just over time squeeze their way, into the matrix, spinning ATP synthase forever, right? Something must be pumping protons into the intermembrane space. Something must be adding the protons in the intermembrane space. Does that make sense? So let's do some more erasing. All right, you guys ready to erase? I'm going to erase a chunk here, and a chunk here, and a little chunk here, and a chunk here. Okay, so I'm doing a little bit of erasing, and now I want to grab a blue marker. All right, you ready to draw some other parts here? There's more parts to this setup here. All right, ready? There's this guy. Okay, I'm drawing this transmembrane protein complex. Here's a different transmembrane protein complex. Here's a different integral membrane protein complex, but not transmembrane. Look how this one doesn't span the entire membrane like the other guys do. And here's another one. This is another transmembrane protein complex. All right, so you see what I drew here. All right. These are called complex 1. This is complex 1, 2, 3, and 4. Okay. Now, let me just tell you what they do first, and then I'll give you more information later. Okay. Guess what these guys do? Complex 1, 3, and 4 are actually proton pumps. Proton pumps. What do you think proton pumps do? They pump protons. It wasn't a trick question. They pump protons. So look at what pump 1 does. This is pump 1. It takes protons from the matrix and it pumps them into the intermembrane space. You see that? It's a one-way pump. Pump 3, same thing. It takes protons from the matrix and it pumps them into the intermembrane space. What do you think pump 4 does? It's not a trick question. It pumps protons into the intermembrane space. You see that? There are 3 pumps that live in the inner membrane of the mitochondria. And by the way, there's thousands of pump 1, there's thousands of pump 3, pump 4, and there's thousands of this guy too, which I'll tell you what he does later. All right? But let me ask you this. if there are 3 pumps, pumps 1, 3, and 4, and their job is to pump protons, they pump protons into the intermembrane space, and that's their job. Would it make sense then why we have a high concentration of protons in the intermembrane space? Because they're pumping those protons faster than the protons are diffusing back in, right? So think about that real quick. Just think about it. There's 3 pumps pumping protons into the intermembrane space. That's what's resulting in the high proton concentration of the intermembrane space and the relatively low concentration of protons in the matrix of the mitochondria. And that's what keeps ATP synthase working, right? So as long as these pumps are pumping, okay, as long as these pumps are pumping, there should be plenty of protons out here to chemiosmose, exert that proton motive force, keep ATP synthase spinning to make ATP. Did you get that? But now let me ask you the next question here. Let me ask Wicket. Wicket, what would be your next logical question about this process, about how it works? What's yours at home? I like that question, Wiki. All right, Wicket just asked if this is active transport of protons. What do you think at home? Is this active transport of protons by these pumps? Or is this passive transport of these protons? Look what's happening. These protons are being pumped from an area of low concentration to an area of high concentration of protons. That's active transport, isn't it? Doesn't active transport need to be powered by something? Okay. So doesn't that mean pumps 1, 3, and 4 need to be powered? What do you think powers pumps 1, 3, and 4? Can you beat Wicket to the answer? No, this is the second time Wicket has ever been wrong. Okay, Wicket, you can't keep saying ATP. It's not ATP. Usually it's ATP, okay, you guys at home. Yes, usually when the professor asks what's powering this process or that process in the cell, the answer is ATP. But actually, it's not ATP in this case. It wasn't ATP for what's powering ATP synthase, and it's not ATP for what's powering these pumps. It's something entirely different. So let me explain that next. All right, so how do these pumps work? To understand, I drew some purple dots here, and what I want you to know is that these purple dots represent cofactors. Usually they contain iron or some other cofactor, but that's not important. What's important is that you understand what these dots do. Each of these purple dots can accept an electron and then pass the electron on. That's their job, right? So what you should know is, do you remember the concept of electronegativity? Do you remember that concept that different elements have different affinity for electrons, and that's called electronegativity? Well, here, what you should know is that the electronegativity of this cofactor here is the lowest, right? This dot right here on complex 1 has the lowest electronegativity, and the one next to it has a higher electronegativity, and the one next to that has a higher electronegativity, higher, highest, okay? So these dots accept electrons and can pass the electrons on, and these dots span, they range from low electronegativity on complex 1 to high electronegativity on complex 4, okay? So let me ask you this. Let me give you an example. This is an example I give my students sometimes. You know, if you go up to a soda machine and you feed coins into that soda machine, those coins go on a journey, right? Those coins kind of go on a journey into the vending machine, right? If you want a soda or you want to buy a can of Pepsi or something, those coins go on a journey. Just like those coins go on a journey, you can feed electrons into this machine. this whole complex forms a machine. Electrons can be added here, okay? Electron coins can be added here at complex 1 or here at complex 2, okay? So you can feed this machine electrons. And so just like you can add coins to a vending machine, you can think of it as electron coins, right? This is an electron coin, this is an electron coin. You can add electrons to this machine. You can add them either here at complex 1 onto this cofactor or at complex 2 at this cofactor. What do you think happens next? Let me ask you that. If I give this cofactor an electron and its neighbor, let's say this neighbor is greedier than this one, what do you think happens next? What do you think, Wicket? Right. The neighbor's greedier for the electron, so it'll steal the electron. You see that? So I fed the machine an electron here. Its neighbor is more electronegative, and it took the electron. Its neighbor is more electronegative, so what do you think is going to happen next? That's right, the electron will hop here, and then the electron will hop here, right? And what if I fed the machine here? Well, this is where this electron would begin its journey. Its neighbor would steal the electron. Its neighbor would steal. Do you see how these electrons are going on a journey? And why do you think they're going from dot to dot? Well, remember, because this final dot right here, this cofactor is the greediest one. It's the one with the highest electronegativity, the highest affinity for electrons. Your book may actually call this standard reduction potential. Your book may not refer to it as standard reduction potential, but just in case your book does, that's what it means. On the left, you have low standard reduction potential, and on the right, you have high standard reduction potential. That's the same way as saying low electronegativity, high electronegativity. If complex 1 is low in electronegativity, well then complex 2 is going to steal its electrons, then complex 3, which is even more electronegative, is going to steal those electrons, and then complex 4 is going to steal those electrons. So that's how it works, right? Imagine a piano, right? Imagine a piano, and each key of the piano from left to right is more and more magnetic, right? A stronger and stronger magnet. So if I have a piano, the key on the left would be the least magnetic, then the one right next to it would be more magnetic, and then next to it would be more magnetic. What would happen if I put a steel ball on the left key and let go? What do you guys think would happen? What do you think would happen if I put a steel ball on a piano key on the left? Wouldn't it zip to the right? Wouldn't that steel ball zip to the right, right? That's exactly what's happening here, you guys. So, but not with magnetism, obviously, but with electronegativity. I'm feeding this machine electrons. The electrons are going on this journey, and they end here on complex 4. I can feed the electrons here, and it goes on the journey, or I can feed the electrons here, and it goes on a journey. And let me ask you this. Is this a spontaneous journey for these electrons? Are these electrons zipping on their own, or is something forcing them to go? Wicket has it right. These electrons are spontaneously zipping through the system from complex 1 or 2 all the way through complex 4. These electrons are moving spontaneously. It's driven by electronegativity, right? So just like if I put a steel ball on the magnetic keyboard and it zips off to the right because the right key is the most magnetic. If I put these electron coins in the machine, they will zip to the right because because of electronegativity. Your neighbors keep stealing you, right? So this is a spontaneous flow of electrons. What I'm trying to get at is this. Do you guys remember energy coupling, right? Energy coupling. This means that you can use the energy from a spontaneous process to power a non-spontaneous process. You remember that? So look, the fact that the electrons are spontaneously moving through this process, right? The electrons are moving through all these complexes. That's spontaneous. That means it's exergonic. That means it releases energy. And that's the energy that's being used to do what? To actively pump the protons. So, let me ask you again. This is active pumping of protons, isn't it? Aren't these pumps pumping protons actively? Does active transport require energy? Where's the energy coming from? Think about it. Can you beat Wicket? From the electron flow. Wicket, you got it. You've totally redeemed yourself at this point. The electrons are flowing. The electrons are flowing. That's what's providing the spontaneous energy. That's what's providing the energy to pump the protons. Isn't that neat? The energy released from flow of electrons is being used to actively pump the protons. Isn't that neat? So I hope that makes sense. This is how this complex works. And now let me name it. I haven't given it a name yet, have I? Let's do this. So now let me give you the name of complex 1, 2, 3, and 4. Together they have a name. I don't just call this complex 1, 2, 3, and 4. I call it the electron transport chain. The electron transport chain. You may have heard of this before, shortened ETC. So when I say the ETC, I mean the electron transport chain. And that's now I hope you understand the role of this little guy, right? He's not a pump, is he? Complex 2 of the electron transport chain is not a pump, is he? But isn't he required as, part of the wire per se? You know why I call this a wire? You know how wires work in electricity? Electrons are used to propel, the electricity. Electrons flow through a wire, right? Well, here, this is almost like a biological wire. Isn't that neat? This is electricity, isn't it? Electrons flowing through a biological wire. Isn't that cool to think about it that way? So the electron transport chain is this complex I've just been talking about. But let me tell you something before we take a quick little break time with Gizmo and Wicket and see what they're up to. Let me tell you something. I personally don't like that name for this complex. Electron transport chain. I really don't like that name. And I'll tell you why. Because look at what it's called. It's called the electron transport chain. That makes it sound like the job of complex 1, 2, 3, and 4 is to transport electrons. Do you think that's its job, or do you think that's how you power it? Wicket, you're on top of it. That's just how you power it, right? But what's the job? Let me ask you this. What's the job of the electron transport chain? Is it to transport electrons, or is it to transport protons? That's right. Its main job, look at you guys, its main job is to pump these protons actively. You see? Look at this. The main job of the ETC is to actively pump the protons so that you have a high concentration of protons so that they can chemiosmose, provide a proton motive force to spin ATP synthase to make ATP, right? That's the job of the electron transport chain. So if I was naming this thing, I would call it the proton transport chain, okay? I would call it the proton transport chain because that's its job. It's like calling a car a gas guzzler. Sure, it guzzles gas, but that's not its job, It's an automobile, right? So, calling this complex 1, 2, 3, and 4 an electron transport chain is akin to calling a car a gas guzzler. Does it guzzle gas? Sure. But is that its main job? No, it's a side effect of doing its job. It's a necessary evil of doing its job, right? So the electrons flow, sure, but that's what powers these pumps to pump the protons. So I can make ATP. All right, you guys ready for break time with Gizmo and Wicket? Now, here's something important that you need to understand. I told you that you can add electron coins to this machine. And what do those electrons do once I add them to the machine? That's right, they zip down from purple dot to purple dot, right? These are cofactors, from cofactor to cofactor, and then they come to a rest here. The electron ends here, right? The electron is now stuck on the last cofactor on complex 4. Well, let me tell you something very important, you guys. Here's the thing. I need a way of removing that electron from this final dot, right? I need to get the electron off the ride. So think about it this way. If you're at, Disneyland and you're on a roller coaster, when you go on a ride, you need to get off the ride at the end, right? If you don't get off the ride at the end, then other people can't go down the ride. Does that make sense? So think of it like the freeway. If people aren't exiting the freeway, well, then pretty soon it gets all jammed up, like a traffic jam, and the whole thing kind of shuts down, right? Like your favorite freeway in your city, okay? So if I can't take this electron off the ride at the end of his ride, then more electrons can't come down the ride, because it just gets, like the electron gets stuck here, then the next one gets stuck here, right? And then the next one gets stuck here, and the next one gets stuck here, and soon they're all, they all have electrons, and there's no more electrons that can join the ride, right? So we need a way of getting off the ride. So we need a molecule that is very electronegative, that likes electrons, right? That means electronegative, to steal the electrons from this dot. If you're going to steal the electrons from this cofactor right here, you need to be pretty electronegative yourself, right? Because remember, complex 4, this is a very electronegative complex. So if you're going to steal from complex 4, you'd best be pretty electronegative yourself. And look at the equation. I want you to look at the equation for cellular respiration. What up there is very electronegative? Oxygen. Great, oxygen. Oxygen is highly electronegative. Remember, oxygen appears at the top right of the periodic table of elements, and this means that it's a highly electronegative element. So that's exactly what happens. Watch this. Oxygen comes along, and do you know what your textbook refers to it as? Not just O2, but 1/2 O2. You know what 1/2 O2 means? Half of an oxygen molecule, so half of an O2, which essentially means what? An O, an oxygen. An oxygen comes along, and it actually picks up that electron. Actually, it could pick up 2 electrons, as well as 2 of these protons, 2 protons. And what do you think that forms? If one oxygen picks up 2 electrons and 2 protons, and remember, a proton and an electron is a hydrogen, and one oxygen picked up 2 electrons and 2 protons, what did it form? Can you beat Wicket? It formed water, H2O. Isn't that neat? So look at this. Oxygen comes along at the end of the ride, at the end of the electron transport chain, or ETC. Oxygen comes along and it picks up 2 electrons from the ride, as well as 2 protons from the matrix, to form what? To form water, okay? To form water. Oxygen picked up electrons to form water. Now, because of oxygen's role as the final acceptor of the electrons, it has a special name. I need you to remember this terminology, this name, okay? Oxygen is known as the terminal, which means final, terminal electron acceptor. The terminal electron acceptor, also sometimes called the TEA, the terminal electron acceptor. It's called the terminal electron acceptor because it picks up the electrons at the end, right? Isn't that neat? And here's something else that's more neat, okay? Since you've been born, you've had to breathe, right? Since you were a baby, you were just born, you've breathed in, right? You know, breathing in, nice and relaxing, but necessary for life, right? Have you ever wondered why you need to breathe? No, really, seriously, everyone knows you have to breathe, right? And I ask my students all the time, why do you need to breathe? They say, to stay alive. Well, why? What's in the air that you need to breathe? They say what? Oxygen, right? Oxygen. And then I say, why do you need oxygen? Then the answer is invariably to stay alive, okay? But no one knows any deeper than that unless you've studied science, unless you've studied cellular respiration. Let me ask you now that I've explained some things to you, let me ask you now, why do we need to breathe? Why do we breathe in oxygen? That's right, because we need someone to take the electrons off the ride, okay? We need a terminal electron acceptor. We need something that's highly electronegative. Oxygen checks that box. We need something that's highly electronegative to pick up the electrons at the end of the ride, at the end of the electron transport chain. And by removing those electrons from the electron transport chain, we make more room for more electron coins to come in and go zip down the train, right? So this electron can add, and it goes down the ride, and then it gets picked up by oxygen, and then this electron goes down the ride, and it gets picked up by another oxygen, and I can keep feeding this vending machine coins. I can keep feeding it electrons. I keep feeding it more and more electrons because I know oxygen's on the other side picking up those electrons, allowing me to feed more machines. But let me ask you this. What would happen if I ran out of oxygen? What would happen if I ran out of oxygen? Yeah, exactly. That wouldn't be good, right? Because if I ran out of oxygen, the electrons wouldn't have a way of leaving this ride, which means the electrons would back up, right, on this machine. The electrons would back up like a traffic jam, and this whole thing would shut down. And that's by the way, some foreshadowing. That's how we branch into fermentation, which I'll explain later on. But as long as we have oxygen and we're breathing nicely, we should be just fine. And this machine works just fine. All right, you guys, I hope you're following along. Try to think of this as a story. Try to use a flow of thoughts, okay? And just try to imagine everything we learned from the beginning, right? What's going on in the mitochondria, ATP synthase. It's powered by protons. The electron transport chain pumps those protons. All right, so let's take a little step back at this point and look at the equation. Look at the equation for cellular respiration. What have we discussed so far? Haven't we discussed oxygen? Haven't we discussed oxygen? And what happens to oxygen during this process? Remember, oxygen picks up electrons to form water, right? So oxygen became the product water. Now, let me ask you this. This is important. Do you all remember the definitions of oxidized and reduced? Remember Leo says grr? Remember that mnemonic device I had you learn? Let me ask you, and let me see if you can beat Wicket. During this process of cellular respiration, is oxygen getting oxidized to water, or is oxygen getting reduced to water? I need you to think about that. That's right, Wicket, you got it. Wicket got it, it's reduced. Remember, when you're talking about the terms oxidized and reduced, you really need to know those terms to understand cellular respiration, because that's all cellular respiration is, by the way, a series of what's called redox reactions. Reduction and oxidation, that's what redox stands for. And that's all cellular respiration really is, okay? So remember what I told you to remember, that little story of the Leo says grr? Leo, L-E-O, Leo, like a lion, says grr, right? G-E-R. Leo stands for loss of electrons as oxidized. Gur means gain of electrons is reduced, right? What is oxygen doing at the end of the ride? Oxygen is picking up electrons, right? So is oxygen gaining electrons to become water? That means oxygen is getting reduced to water. So here you can say oxygen becomes reduced. You should know that during cellular respiration, oxygen, your terminal electron acceptor, is getting reduced to water, the product. Okay? And again, I can't stress to you how cool this is, you guys. Now you finally know why you've been breathing your whole life. This is literally the reason you're breathing in. You know, when you were born and you needed to breathe, okay, this is why. This is why. The cellular respiration is why. Because students ask all the time, why do we have to learn this convoluted thing, you know, with all these parts and this electron transport chain, the ATP synthase and protons and what in the world, you know, but it's vital. It's literally vital that you understand this. You know why I say literally vital? Because you know, that's your vitals. Your vitals include, are you breathing, right? So breathing in oxygen your whole life, the reason for that has been you need oxygen as a terminal electron acceptor to pick up the electrons, so the oxygen gets what? The oxygen gets reduced to water. That's why you breathe. That's why you need oxygen, okay? That's literally why you breathe. So let's say you want to be a doctor in the future, or a nurse in the future, or any scientist or clinical person, or even if you want to know why people breathe, right? You need to know your cellular respiration. That's how important this topic is. It's not just there to, you know, stress you out with how convoluted it is. It's there because it's vital. It's very important that you understand, you know, how it works. You know, if you want to go in anything medicine related, you should explain why, you know, you need oxygen to live. And this is why people need oxygen to live. So this is vital to understand in more than one way, right? No pun intended. Actually, puns intended. All right, so let's keep going. Let's keep going. So we have explained so far why oxygen is required. It's your terminal electron acceptor, which gets reduced to water. Okay, great. But we have not explained the role of glucose, and we have not explained the role of CO2, or, you know, and we have explained 28 of these ATP, but not all of them. Okay, so we still got some things to explain, right? So let's turn our attention now to glucose. I want you to take a look at glucose. Glucose has several hydrogen atoms on it. It has several hydrogens. And so do fats. Do you know fats? We learned about fats. Here's a picture of a fat. Okay. Remember, fats have these long fatty acid tails, these 3 long fatty acid tails. And what are those tails just full of, just clamoring with? Tons and tons of hydrogens, right? So fats have hydrogens on the tails. Sugars, like glucose, have hydrogens. And so why do I keep talking about hydrogens? Well, that's because this is what we need. This is why these are energy molecules. Sugar has energy because of its hydrogens. Fats have a lot of energy because of the hydrogens. Proteins have a little bit less energy, and it's because they're lacking a lot of those hydrogens, right? But it's these hydrogens I want to talk about. Let me ask you this. Look at this. Remember, what did I say over there? What is a hydrogen? What is a hydrogen atom? Remember, a hydrogen is simply a proton. A proton, what's another way of writing proton? H+. Hanging out with what? A hydrogen is a proton hanging out with an electron. That's all a hydrogen is, isn't it? A hydrogen is a proton hanging out with an electron. And believe it or not, this is why sugars and fats are so full of energy. Because let me ask you this real quick. I just taught you this process here. You see this here? All this stuff going on? What powers this whole machine? What powers the machine? Isn't this whole thing powered by electrons and protons? Think about it. Electrons flowing powers protons pumping, which then results in ATP. So do you agree that it's nothing more than electrons and protons that power this powerful machine of the electron transport chain plus ATP synthase? That's all you need to power this machine to give you this awesome amount of ATP, 28 ATP per glucose. Now look at this. Sugars and fats are chock full of hydrogens. Hydrogens have the protons and the electrons necessary to power the powerful machine. You putting 2 and 2 together? What gives you more energy? What gives you more energy, sugars or fats? Think about that. Can you beat Wicket? That's right, Wicket. Fats, right? Fats. Why do you think fats give you so much more energy than sugar? Based on your understanding that hydrogens power this machine. Wicket, you're on top of it. It's because fats have so many hydrogens on those fatty acid tails that each one of those hydrogens, you know what you want to do with those little hydrogens, those yummy little hydrogens? You want to pluck them off, and feed them into the machine. So that's what we do, you guys. That's how cellular respiration works. You take sugars or fats, and you pluck each hydrogen off. So what I want to do with glucose, you know what I want to do with glucose? I want to pluck each and every one of these hydrogen off of glucose, and I want to take it over to the machine so I can power the machine. I can feed the electrons through the electron transport chain, and the protons can get pumped out, right? You see what I'm saying? You follow me? What I want to do is take sugars and fats. I want to take the hydrogens, and I want to feed the hydrogens into the machine. That's how I'm going to power the machine. Now, let me ask you this. If I were to take all the hydrogens away from glucose, right, C6H12O6, if I were to take all the hydrogens away, what's left? Just take a look. I want all the hydrogens to feed into the machine. If I take all the hydrogens over to the machine, what's left? CO2. So do you see what happens here? I take all the hydrogens away from glucose, and all that's left are C's and O's, and that forms my product, CO2. Isn't that interesting? That's why CO2 is a product. Because when I take glucose and I remove all the hydrogens, because I want to feed the hydrogens into this machine to power the machine, all that's left of my glucose are the C's and the O's, CO2, okay? Now let me ask you this. If I take H's away, and remember H's also contain electrons, right? If I take the H's away from glucose, have I reduced glucose to CO2, or have I oxidized glucose to CO2? Think about that. Can you beat Wicket? That's right, Wicket. I have oxidized. Remember, oxidized is Leo. Loss of electrons means oxidized. I have removed hydrogens. That means each hydrogen contained an electron. I have removed electrons from glucose. So I have oxidized. You see here, look. I have oxidized glucose to CO2. Okay, very important that you understand that during the process of cellular respiration, glucose is oxidized to CO2, while oxygen is reduced to water. Very important for you to understand that. And how does your body, here, think about this. When you breathe in, you mostly want what? You want oxygen, right? And why do you need oxygen when you breathe in? To live, right? You're like, to live. But why do you really need oxygen when you breathe in, based on what we just learned? Because you need the terminal electron acceptor, right? Oxygen gets reduced to water. Great. But let me ask you this. When you breathe out, what do you breathe out a high concentration of? You know this, right? What do you breathe out more than you breathed in? Everyone knows you breathe out CO2, right? You guys know that. Why do you breathe out so much CO2? Well, think about it. What's left of your glucose, after I've oxidized the glucose, what's left of glucose? CO2. So that CO2 leaves my cells, it gets into my blood, it goes to my lungs, and I breathe out the CO2. So isn't that interesting? The reason you've breathed in since you were born is because you need oxygen as a terminal electron acceptor. And the reason you've been breathing out so much CO2 since you've been born is because that's what's left of your glucose and your fat molecules after you have fully oxidized them, right? That you've broken them down with cellular respiration. So I'm going to blow your mind again real quick, if you don't mind, unless you mind getting your mind blown, right? But I'm going to blow your mind again. And I'm going to tell you that when you lose weight, when you exercise, and you lose weight, let's say you've been exercising for a month and you've lost a couple pounds, right? You've lost some weight. You lost 2-3 pounds. You've been exercising that month and you've been trying to be good about your diet, and you've lost a little bit of weight, so you're trying to get in shape, right? Where did that weight go, right? You ever thought, where did that weight go? Did it disappear? Did I sweat it out? Did I poop it out? Did I pee it out? Sorry to be a little gross there, but where did it go, right? People don't know. Most people don't know. Well, believe it or not, a majority of that weight left your body through your mouth, through breathing out. Isn't that neat? Think about it. Look at this. Weren't these the sugars that were weighing down your body? Sugars weighing down your body? And after you've oxidized it, what's left? CO2, which leaves your body how? By breathing it out. And what about fats? Remember fats. When you break down fats, what's left of the fat? Also CO2, right? Also CO2. So those fats that are weighing down your body, right? Those fats that make up your, that fill up your fat cells, right? Those fats that are causing you to not like the number on the scale sometimes, right? Those fats leave your body as CO2 as well. So when you lose weight, you're losing the majority of your body's weight just by breathing out. Okay, and now don't try just sitting there and breathing really fast. That doesn't work. I know what you're thinking. What if I just sat here and breathe really fast? Would I lose a bunch of weight? Actually, that doesn't work at all, right? Because all you're doing then is sucking in air and blowing out the same air. Your body has to produce more carbon dioxide, right? How could you get your body to produce more carbon dioxide? Well, you would have to exercise, right? Because when you exercise, your body needs more ATP, to keep going, right? Because ATP powers your cells, correct? So when you're exercising, your body needs more ATP. What's the only way to get more ATP faster? Well, it's to break down more sugar and more fats and use more oxygen. This is why you start breathing so hard. When you exercise, you start breathing really hard. You need more oxygen to burn more sugar and more fat faster, right? And then when you blow out, when you exhale, you actually exhale much more CO2 than you normally do, right? And to couple that with a diet, why do you diet? If you want to lose weight, why do you diet? Well, it's to make sure that you have fewer sugars and fats to burn. So do you see why diet and exercise go hand in hand for losing weight? Because with exercise, you're converting more sugars and fats to ATP, CO2, and water, CO2 which you breathe out. And with dieting, you have fewer glucose and fats to burn. So by having fewer fats to burn and burning them faster, you're going to exhale more CO2, right, than the amount of, sugars and fats you consume. So why do I say all this? Well, because some of you want to study nutrition, or you want to go into exercise physiology. You cannot understand nutrition. You cannot understand exercise physiology, any of this stuff without understanding cellular respiration. Because if you don't fully understand cellular respiration, you don't understand how weight actually leaves your body, right? Through CO2. You don't understand how diet and exercise truly work unless you understand cellular respiration. So when students ask, why do we have to learn cellular respiration? It seems kind of archaic and boring. Well, remember, if you want to be a competent person in medicine, you want to explain to a patient why they're breathing. You want to explain to a patient why they're breathing out CO2. You want to explain to a patient the benefits of diet and exercise and why they're important and how people gain weight or lose weight. You cannot competently do that without understanding cellular respiration. Isn't that neat? So let's keep going. All right, at this point, I have a kind of a confession. What I've been doing so far is explaining cellular respiration backwards. I said at the beginning of this video that I have my own special way of explaining cellular respiration, and this is the way I've come up with. This is the way I've been teaching cellular respiration for over 10 years now, and it's backwards. I start with the end of the story, and I work my way towards the beginning of the story because it's just easier to understand this way, okay? Everything I've explained so far, remember, everything I've explained so far includes the electron transport chain, complex 1, 2, 3, and 4, ATP synthase, which spins and makes ATP, all this stuff right here, which the electron transport chain plus ATP synthase, chemiosmosis, this is called oxidative phosphorylation. Okay, oxidative phosphorylation. That's the process I have spent some time explaining to you on the board. And guess what? Cellular respiration has 3 major phases. And this is phase 3, okay? This is phase 3. So you now understand phase 3 of cellular respiration. But guess what? There's two and a half other phases, okay? There's phase 1, which is called glycolysis. There's phase 1.5, okay, this is phase 1.5, and phase 1.5 is called oxidation of pyruvate. And then there's phase 2. Phase 2 is called the citric acid cycle, also known as the Krebs cycle, also known as the TCA cycle. Your textbook may call phase 2 the citric acid cycle, or it could call it the Krebs cycle or the TCA cycle. Don't get confused. They all mean the same thing. I think our current book, Campbell's 12th, uses citric acid cycle, so that's what I'm going to be going with. But remember, citric acid cycle is the same as Krebs cycle or TCA cycle. So now you might be, you might feel deflated. You may feel like, oh my gosh, like, wait, all this explanation with all these colors on this board, which it looks convoluted, all this stuff was just part 3, and it's true. But there's a reason I start with part 3, and I just want to share that with you now, okay? Look at this. Part 3 is so important, right? You see how ATP synthase works, and how many ATP did ATP synthase make? 28 for every glucose, right? So look, 28 of these 30 net ATP are made right here. 28 of these net ATP are made right here. So that's the most important part of it, the cellular respiration, is ATP synthase making this lion's share of ATP, 90% of the ATP, the 28 ATP. The rest of these phases, you see glycolysis, oxidation of pyruvate, citric acid cycle, these don't make tons of ATP, okay? In fact, look, glycolysis gives you a whopping 2 ATP, okay? Glycolysis gives you a whopping 2 net ATP. And the citric acid cycle gives you a whopping 2 net ATP, okay? So you get 2 ATP here during glycolysis. You get 2 ATP here in the citric acid cycle, but who cares? That's nothing compared to the 28 ATP that's made here, right? And by the way, let me tell you something real quick, okay? I forgot to define something. The ATP that's made by ATP synthase with the help of oxygen during oxidative phosphorylation, this ATP is made by what they call OXPHOS, or oxidative phosphorylation. So if an exam question asks, by what process does ATP synthase make ATP? This is the 28 ATP that are made here were made by oxidative phosphorylation. That's the process by which these ATP were made. Any ATP made during oxidative phosphorylation, this process by ATP synthase, those 28 ATP were made by what's called oxidative phosphorylation, which means that these 2 ATP and these 2 ATP are not produced by oxidative phosphorylation. They are produced by something called substrate level phosphorylation, which I'll explain in a bit. And these 2 ATP were also made by substrate level phosphorylation. Okay, great. Now, again, the lion's share of ATP is made at the very end by ATP synthase, and a piddly amount is made during the citric acid cycle and glycolysis. So let me ask you this. If you've been paying attention, okay, I know you've been paying attention at home. I know Wicket's been paying attention, unless he's napping. Let me ask you this. What do you think is the main point of glycolysis, oxidation of pyruvate, and citric acid cycle? What do you think is the main point of those steps? If it's not directly to make ATP, then what do you think those steps focus on? What happens during those steps? Just use logic and reasoning, right? Just use logic and reasoning. What do you think is going on during those early phases? Glycolysis, oxidation of pyruvate, citric acid cycle. Think about that. Can you beat Wicket? Wicket's about to chime in here. That's right, Wicket. The whole point is to oxidize glucose. Now let me tell you something. Look at this. Remember, what did I say? What did I say is the main point of, cellular respiration? To make ATP using what? Look, glucose. What do we want from the glucose? What do we want from the glucose? Do we want its carbons, its hydrogens, or its oxygens to power this machine? Which one do we want? We want these hydrogens, right? Well, let me tell you something. Taking 12 hydrogens from a glucose molecule is not easy. You can't just take all 12 of these hydrogens in one step, okay? Let me put it this way. If you could take these 12 hydrogens from glucose in one step and get them all to the electron transport chain and get them all to the final step in one step, then there wouldn't be a phase 1, a phase 1.5, and a phase 2. Because you know why? It turns out it takes many different steps. Remember, a metabolic process takes many enzymes, right? There's a metabolic process that takes many, many steps, right? Well, it turns out it takes a lot of steps and a lot of enzymes to fully oxidize glucose to CO2, to take each and every one of these hydrogens away from glucose and get those hydrogens over to the electron transport chain. Does that make sense? And that's the whole purpose. That's the whole reason you have glycolysis. That's the whole reason you have oxidation of pyruvate. That's the whole reason you have the citric acid cycle. It's to fully oxidize your glucose. So during glycolysis, you start with glucose, right? You start with glucose. You start with glucose. And what did I say you're left with at the end of cellular respiration? Your glucose gets fully oxidized to CO2. So by the end of the citric acid cycle, all that's left of your glucose is CO2. Okay, so you have fully oxidized your glucose. And here's the thing. What you want to do is take the H's, from glucose and you want to feed them to the machine. And here's how you do it. There are two electron carriers. They're called NAD plus and FAD. These are electron carriers. This is what NAD plus looks like. It's a dinucleotide. You don't need to know this for my class, but NAD stands for nicotinamide adenosine dinucleotide. Here you can see the molecule. It's a dinucleotide, and here it is in its oxidized form, okay? And what NAD does, what NAD plus does, is it comes over here to one of these early steps. It picks up two electrons. And a proton. Okay, it picks up two electrons and a proton to become what? To become NADH. And then it delivers that H to the electron transport chain. It gives the H to the electron transport chain. NADH gives its H to complex 1. Its electron goes through complex 1. Its proton pops off, and it's one of the protons that gets pumped across the membrane. Another thing that's another of these electron carriers is FAD. FAD can pick up two electrons and two protons, and FAD gets reduced to FADH2, which then goes and drops off its electrons at complex 2. Okay, so here's I know it's a little confusing, but here's how it works. During glycolysis, oxidation of and citric acid cycle, right? Oxidation of pyruvate and citric acid cycle. During these steps, H's are taken from the sugar and handed to the electron carriers, either NAD plus or FAD. These carriers, once they pick up those H's, they get reduced to their reduced form, right? So NAD plus picks up some H's to become NADH. FAD picks up some H's to become FADH2. And those H's travel to the electron transport chain, and NADH visits complex 1, right? It's what feeds the coins, the electron coins, into complex 1, while FADH2 visits complex 2, and it feeds its little electron coins into complex 2, right? And those are the electrons that zip down the electron transport chain to be picked up by oxygen at the end, right? To pump the protons to power the ATP synthase. Isn't that neat? So the H's on glucose, are given to either NAD plus or FAD. That reduces these oxidized carriers to the reduced form. The carriers then carry, that's why they're called electron carriers. They're called electron carriers because they carry the electrons from glucose to the electron transport chain. And I know that's kind of a misnomer too, because students ask, but they're carrying H's, they're not carrying electrons. But remember, electrons are part of a hydrogen. Sure, NAD picked up 2 electrons and a proton, and FAD picked up 2 electrons and 2 protons. So technically they're hydrogen carriers, and if I could rename these guys, I would call NAD plus and FAD hydrogen carriers, because what they're really doing is taking hydrogen from the sugar and carrying those hydrogens to the electron transport chain. NADH drops off the hydrogen here, FADH2 drops off the hydrogen here, but they call them electron carriers because the more important cargo is actually the electrons, because the electrons are what's powering the electron transport chain. The protons are just getting kind of pumped across, but they would kind of be there anyway, okay? So that's why they're called electron carriers. But if I were to rename them, I would call them hydrogen carriers, right? But these are the two electron carriers. So this is the whole point, you guys, right? Listen to this. This is the whole point of steps 1, 1.5, and 2. During glycolysis, you're going to take your glucose and you're going to steal some of its H's, right? You're going to steal some of those H's. And when you steal those H's, you're going to give them to NAD+. You're going to create 2 NADHs, right? That means you have given some of those hydrogens to NAD+, reducing it to NADH, okay? And where do you think NADH goes once it's created? Right? So I stole some H's from glucose, okay? And now I have NADH. Well, the NADH travels to the complex 1 to drop off the H. You see what happens? And when it drops off that H, it becomes NAD plus again, and it can come back and pick up more electrons from another glucose molecule. So that's what these carriers do. During glycolysis, glucose gives a couple of H's to NAD+, making NADH. NADH books it over to the electron transport chain. It feeds the electrons into the electron transport chain and the protons get pumped, okay? And then NAD+ is reformed. It comes back. It picks up more electrons and does it over and over again, okay? And that's great because that's going to power these pumps to pump the protons to make ATP. Does that make sense? That's the important part of what's happening here. And then at the end of glycolysis, we haven't fully stolen all the H's. We have not fully stolen all the H's, okay? In fact, here is exactly what's happening during glycolysis. So here we can see an overview of aerobic cellular respiration. Remember, stage 1 is called glycolysis, and we're going to delve into that in just a second. Glycolysis is first stage, followed by stage 1.5. Remember, stage 1.5 is pyruvate oxidation. Next, we have stage 2, the citric acid cycle, also known as the Krebs cycle, also known as the TCA cycle. And then lastly, remember on the board, all that explaining I did, that was oxidative phosphorylation, which includes the electron transport chain as well as chemiosmosis through ATP synthase. So this, I love this slide. You may want to take a picture of this or screen grab of this, because this is an awesome slide that shows a big picture overview of all of cellular respiration. And here you can see all the steps laid out. I just want to show you something, again, before I delve into glycolysis in detail, I just want to show you something. Look how glycolysis is the only one of the steps that occurs in the cytosol, the liquid portion of the cytoplasm. You see this? Glycolysis occurs in the cytosol or cytoplasm. The rest of these steps occur inside of the mitochondria. All right, so that's one thing to understand. And again, remember, glycolysis is the first phase of this process, and we're going to go into that in detail. This is a process where glucose is processed into 2 molecules of pyruvate. We also produce 2 NADH and 2 net ATP. And that ATP was formed by substrate level phosphorylation, which I'll explain more in just a second. So next, let's delve into glycolysis. And again, one more big picture overview before we delve into the details of glycolysis. During glycolysis, the input is mainly glucose, which is then processed through glycolysis into 2 molecules of pyruvate, 2 net ATP, and 2 NADH. Now let me show you step by step how this process works. All right, so here you can see the process of glycolysis. It's a number of steps, as you can see. This is a metabolic pathway with several steps. Now, what I'm going to do is I'm going to explain each step in more detail than you really need. So don't get bogged down in the details, because I'm going through details so that you can see step by step what's going on, but I'm not going to need you to know every little step, okay? First, I'm going to explain it in great detail. Then I'll let you know what you need to remember, right, for the exam, okay? So let's start at the top here. Let's start here, okay? These are the first steps, okay? Let's start at the top here. These are the first steps to glycolysis. And remember, glycolysis occurs in the cytoplasm, right, of the cell. And so you start here with what's called the investment phase. It's called the investment phase because we are going to invest 2 ATP molecules. So let me show you how this works. All right, so again, this is the investment phase of glycolysis, which of course happens in the cytoplasm. You start with a molecule of glucose, and look at this pink structure here. This pink structure has 6 carbons, 1, 2, 3, 4, 5, 6. These 6 carbons represent the 6 carbons of glucose, but obviously glucose isn't just a chain of 6 carbons, right? They didn't draw the whole carbon molecule. They just drew the 6 carbons, okay? But remember, this is C6H12O6. Now, the first thing that happens to glucose is that an ATP comes along, and the ATP gives one of its phosphate groups to glucose. So you see, if ATP gives a phosphate group to glucose, glucose has the phosphate group, and ATP is missing one of its phosphate groups. It becomes ADP. This process is known as phosphorylation. When a phosphate group is added to a molecule, this molecule is phosphorylated. So now you have a phosphorylated form of glucose called glucose 6-phosphate. Why? Because it's still glucose, but the 6th carbon has a phosphate group attached. Do you see how that works? Next, there's an isomerization that occurs where glucose gets its bonds rearranged into fructose, right? Fructose is an isomer of glucose. So glucose was isomerized to fructose. So now it's known as fructose 6-phosphate. Next, that type of step happens again, where another ATP comes along, and this ATP also phosphorylates fructose 6-phosphate. So now you have fructose with 2 phosphates, because each of these ATP donated a phosphate group to the sugar. And now this is a doubly phosphorylated sugar, right? So now it's called fructose 1,6-bisphosphate. Fructose, this is the sugar fructose. It has a phosphate on the 1st and the 6th carbon. That's why it's called bisphosphate. That means 2 phosphates. Okay, so do you see why do you see why this is known as the investment phase of glycolysis? Because 2 ATP were invested. 2 ATP were used up to get the ball rolling. This primes the pumps. This allows the subsequent steps to happen, okay? So this is known as the investment phase. We have invested 2 ATP to get to this point. So here, this is now the payoff phase. What happens is, look up here. Do you remember where we left off? We were talking about fructose 1,6-bisphosphate. Well, now the lysis part of glycolysis occurs. Do you guys know what glycolysis means? Glyco refers to sugar or glucose. What do you think lysis refers to? Remember hemolysis or lysis? What does lysis mean? Remember, lysis means break. So what we're about to do is break the sugar in half. You see right here, we're going to break the sugar in half into two, okay? So now we've broken the 6-carbon sugar into two. Then both of these are called glyceraldehyde 3-phosphate. That means it's a 3-carbon sugar called glyceraldehyde with a phosphate on the 3rd carbon. And here's one glyceraldehyde 3-phosphate from this half of the fructose. And here's the other glyceraldehyde 3-phosphate from the other half. So what have we done? We have taken a single 6-carbon sugar and we've chopped it in half. One half goes down this column, the other half goes down this other identical column. So whatever happens down this column is mirrored in this column, because we have two 3-carbon sugars now, not just one 6-carbon sugar, right? Now, at this point, remember, what do we want from these sugars? Remember what I said at the board? What do we really want from these sugars? What's the yummy component of these sugars that we want to harvest? Do you recall? That's right, Wicket. We want hydrogens, right? So we're going to capture some of the hydrogens from these sugars, and we're going to transfer those hydrogens to the electron carriers. Remember NAD+? NAD+ will pick up 2 electrons and a proton and become reduced to NADH. And so will the identical pathway on the right. NAD+ will pick up 2 electrons and a proton and become reduced to NADH. Now let me ask you this. And see if you were paying attention earlier at the board. What do you think these NADH do at this point? This NADH right here and this NADH right here. What do you think they do once they are formed, once they have captured those yummy hydrogens from the sugar? What do you think they do? That's right. They head right over to the ETC. Anytime you see NADH formed, it makes its way right to the electron transport chain. Okay, and it drops off those electrons at the electron transport chain. And NADH, remember, it drops off its electrons at complex 1 of the electron transport chain. So at this point, the sugar has been oxidized, right? The sugar has lost electrons, right? The sugar has been oxidized, and it's picked up another phosphate group. So now what you have are 2 of these molecules here. This molecule here, it's called 1,3-bisphosphoglycerate. It is a 3-carbon sugar with 2 phosphate groups, one on the 1st carbon and one on the 3rd carbon. And remember, you have 2 of these. You have one here and you have like one over here, right? So because whatever's happening to this glycerate is happening to that one. Now, here is the payoff phase, okay? Remember, you made an investment of 2 ATP, but now we are going to pay off that investment. That investment's about to pay off. All right, so how do we do this? This is a process known as substrate level phosphorylation. Let me show you how it works. If we make ATP by taking a phosphate group like this one, see the phosphate group attached to a substrate such as a sugar or organic molecule, we're going to transfer, we're going to transfer this phosphate group from this glycerate, phosphoglycerate. We're going to transfer that phosphate group to ADP. What do you think would happen if I transfer this phosphate group to ADP? Well, that ADP becomes ATP. And so we've just formed ATP. But this is known as forming ATP by substrate level phosphorylation. This means that the way we made the ATP was by getting a phosphate group from a substrate, from a organic molecule like this. So anytime you see like a substrate, like an organic molecule that has a phosphate group, and that phosphate group is transferred to an ADP to make an ATP, that ATP was made via substrate level phosphorylation. So how many ATP have we made at this step right here? We have made 2. See, we've made an ATP here, and we've made an ATP here. So didn't we just earn back our investment? Didn't we just earn back, we invested 2 ATP at the beginning. Those ATP phosphorylated our sugar, and now we gave it back, right? We gave the phosphate back to ADP to make ATP, and we just made 2 ATP back. We've invested 2 ATP, and we made 2 ATP back. Okay, and then at this point, we still have a phosphate group on both of our sugars, so we move that phosphate group around, and then we give the last phosphate group to an ADP, again with substrate level phosphorylation to make ATP again. So we actually make 2 more ATP. Okay, so how many ATP did we invest? 2. How many ATP did we get back? 4. So let me ask you this. How many ATP do we net? Net means come out ahead, like profit, right? If you invest 2 and you get back 4, your net is actually 2 because you're up 2, right? Okay, does that make sense? So we say that during glycolysis, we net 2 ATP, right? And we formed 2 NADH. But let me ask you this. After your poor glucose has been manipulated and chopped in half and changed around, do you think it's still called glucose? No, at the end of glycolysis, you're going to end up with, remember, with 2 3-carbon sugars, right? 2 3-carbon molecules. These 3-carbon molecules are called pyruvate or pyruvic acid, depending on your textbook, right? And again, how many of these pyruvate do you have? At the end of glycolysis, how many of these 3-carbon sugars should you have? 2 of them, right? Remember, 1 down each of these pathways, because a 6-carbon glucose got broken into 2 3-carbon sugars, and now you have 2 pyruvate molecules. So again, I don't need you to remember each and every little step, okay, of glycolysis. You may need to learn every single step if you're going to go off to, like, let's say med school and you're going to study for the MCATs. There might be some technical questions in there. But for the most part, in my class and many other classes, you really don't need to know every little tiny step. But I explain every step so that you can see kind of what's going on with this metabolic pathway. But what I really want you to know are just the general things. Like, for example, where does glycolysis occur? Let's go back to this helpful slide. Where does glycolysis occur? The cytosol or cytoplasm. Great. How many net ATP did we get? 2. By what process? Substrate level phosphorylation. How many NADH were formed? 2. And where did those NADH go once they were formed? Look at this white arrow. I like this figure. See, look at this. Look at this white arrow. It's showing you that the 2 NADH that were formed by glycolysis are going where? Follow the white arrow. It's headed to the electron transport chain. Remember to drop off that hydrogen cargo, to drop off those electrons at the electron transport chain. The electron transport chain is going to be powered by this H. It's going to be powered by these 2 electrons that are being carried by NADH. Does that make sense? So again, what do you need to know? You need to know glycolysis occurs in the cytosol, and you should know the inputs and outputs. So glycolysis breaks down glucose into 2 pyruvate molecules, 2 ATP, and 2 NADH. All right, welcome back. So now you know exactly what's happening during glycolysis. And remember, by the end of glycolysis, your 6-carbon glucose has now been broken down into 2 3-carbon pyruvates, right? Well, those pyruvates still have hydrogens for us to steal. Those 2 pyruvates are headed towards oxidation of pyruvate. And here, oxidation of pyruvate is going to take those 2 pyruvates. It's going to take these 2 pyruvates, and this is what it's going to do. All right, yes, it is time for step 1.5. Remember, At the end of glycolysis, you formed 2 pyruvate molecules, right? 2 pyruvate, 3 carbon molecules of pyruvate. Now watch. Both of these pyruvate head into the mitochondria. The pyruvate head into the mitochondria. And during this step, this is the step known as pyruvate oxidation, also known as oxidation of pyruvate. Either way, it means the same thing. So let's explore what happens during pyruvate oxidation here. All right, so remember, at this point, your pyruvate is heading from the cytosol into the mitochondrion. And did you only have 1 pyruvate or 2? That's right, Wicket. You have 2 pyruvates, right? So you have 2 pyruvates, 2 3-carbon molecules. See, 1, 2, 3. 2 of these 3-carbon pyruvates are heading into the mitochondrion. And here's what happens as it goes through. When the pyruvate enters the mitochondrion, it gets oxidized, right? So a couple of its hydrogens are taken away from it and given to NAD+, reducing NAD+ to NADH. So again, pyruvate is oxidized so that NAD+ can be reduced to NADH. Remember, NAD+ picks up electrons, so it's reduced. And what happens once this NADH is formed? Where does it go once it's formed? Remember? That's right. It heads off to the electron transport chain. Anytime you see NADH formed like this, it's got the H cargo. It's ready to go to the electron transport chain and drop off those electrons, right? Now, as a consequence, a carbon, you see this carbon? Pyruvate was a 3-carbon molecule. You see this carbon right here and these 2 oxygens? Well, they get cleaved, okay? During this process, the carbon and 2 oxygens are cleaved as carbon dioxide, and they leave the cell as carbon dioxide. By the way, this is some of the carbon dioxide that you breathe out. This becomes carbon dioxide that you exhale. So now, if that happened, if you lost one of your 3 carbons, then how many carbons do you have left over on each of your pyruvate? 2, right? So now look, this is a 2-carbon molecule. Your 3-carbon pyruvate is now a 2-carbon molecule. Now, this 2-carbon molecule picks up a coenzyme. Do you guys remember what a coenzyme is? It's an organic cofactor. This coenzyme is called coenzyme A. And this organic cofactor is added on, and here's the organic cofactor called coenzyme A. So the resulting molecule has 2 original carbons as well as this coenzyme A. It's called Acetyl-CoA. And again, I want you to multiply everything here by 2, right? Because again, did you start with 1 pyruvate or 2 at the end of glycolysis? 2 pyruvates. That means that in this step, you form 2 CO2s, 2 NADHs, which head to the electron transport chain, and you form 2 acetyl-CoAs. 2 acetyl-CoAs are left over, right? Again, during this step, what was this step called? Oxidation of pyruvate. 2 pyruvate enter the mitochondrion. 2 CO2 are formed, 2 NADH are formed, and 2 acetyl-CoA are left over. Let's go back to our nice little overall sheet here, this summary sheet, and check it out. Again, look at this is why I love this summary sheet. Look at this. 2 pyruvate enter the mitochondrion and become 2 acetyl-CoA. In the meantime, 2 NADH are formed, which go where? Follow the white arrows. They go to the electron transport chain to drop off those H's. 2 CO2 are formed. Remember, look at, you started with glucose, which is a 6-carbon molecule. Well, 2 of those carbons just left as CO2. All right, and what's left over? Acetyl-CoA. At this point, you have 2 acetyl-CoAs left over. This is what's left of your original glucose molecule. Your original glucose molecule is now 2 acetyl-CoAs. And those acetyl-CoAs are on their way to the citric acid cycle for further oxidation. Now, welcome back. Now you know that during oxidation of pyruvate, this half step, 2 pyruvates are broken down into 2 acetyl-CoA. Along the way, remember, 2 NADH are formed and 2 CO2. CO2 is going to leave your body as, air. And then the 2 pyruvates are broken down into acetyl-CoA, and you form 2 NADH, right? Where did the NADH go that are formed during this step? That's right. They're going to go to complex 1 to drop off their electrons, okay? And what do we have left? 2 acetyl-CoAs. Now, these 2 acetyl-CoAs, they still have yummy hydrogens, right? They still have hydrogens for us to feed on, okay? Those 2 acetyl-CoA are headed to the next step, the citric acid cycle, aka the Krebs cycle, aka the TCA cycle. And here is how that cycle works. So at this point, pyruvate oxidation has been completed. 2 pyruvate have been converted into 2 acetyl-CoA. And remember, both of those acetyl-CoA molecules are headed for the citric acid cycle, also known as the Krebs cycle, also known as the TCA cycle. All right, so let's go into detail about this cycle. During this cycle, you're going to see, and again, I'm going to break it down step by step for you, but you're going to see that during this cycle, we are going to oxidize our acetyl-CoA. We are going to get 6 NADH formed, 2 FADH2s, 4 CO2s, and 2 ATP via substrate level phosphorylation. But let's go break it down step by step so we understand it fully. All right, here it is, the citric acid cycle. Check it out. Here at the top, you have acetyl-CoA entering the cycle, all right? And this is a metabolic cycle. Now, real quick, before I get started with this cycle, I want to ask you, how many acetyl-CoA did we have at the end of the previous cycle? Was it just 1 acetyl-CoA, or did we have 2? That's right, Wicket. We had 2 acetyl-CoA. That's awesome. So, because remember, our glucose was processed into 2 pyruvates, and then those 2 pyruvates were converted into 2 acetyl-CoAs. So remember, this is what I need you to understand. This is showing you in this figure here, you're seeing what's happening to 1 of the acetyl-CoAs. But if you want to know what's happening overall, you have to understand that you're going to get 2 rounds of the citric acid cycle because you have 2 acetyl-CoAs, and this cycle is simply showing you what's happening to 1 of the 2. So you have to multiply everything here by 2 to get the full picture, okay? So just with that in mind, these 2 acetyl-CoAs are going to enter the citric acid cycle. Okay, but what happens to each acetyl-CoA? Now watch this. Okay, I want to show you something very interesting. When you look at a biochemical cycle like this, you see how it's a cycle? A biochemical cycle is called a cycle because the thing that's made at the end, what's made at the end here, I'm circling it. You see this guy right here? This is called oxaloacetate or oxaloacetic acid. Sometimes your textbook might call it that, but this is oxaloacetate. And this is the organic molecule that needs to be replenished. It gets replenished. Replenished means remade at the end of the citric acid cycle. And why is that so important? Well, because the thing that's made at the end of the cycle is needed to start the cycle again. That's what a biochemical cycle is. The product of the cycle is needed to start the next cycle. So what happens? Let me show you something neat. your acetyl-CoA, which has 2 original carbons from your glucose, as well as this coenzyme A, this acetyl-CoA enters the cycle and it joins forces with oxaloacetate. So literally, they get bound together. So oxaloacetate, this organic molecule, binds covalently to acetyl-CoA, and in this process, you lose the coenzyme A. Coenzyme A comes off, and now you've actually combined acetyl-CoA with oxaloacetate. And now you have this mega molecule here. Well, it's not so mega, but you get the picture. It's a bigger molecule, isn't it? Because this is what's left of your acetyl-CoA, and this is what's left of oxaloacetate. This is a big molecule called citrate. And by the way, I'm going to tell you something really neat and interesting just now. Citrate is another word for citric acid, right? Citrate is citric acid. And this is why this is called the citric acid cycle, because the very first thing you do is actually make some citric acid by combining what? Acetyl-CoA with oxaloacetate. So one thing I do want my students to know, I do want you to know, is what needs to be replenished at the end of the citric acid cycle. The answer would be oxaloacetate, and to make citric acid. Very interesting, right? Now, just to take a step back, what do you think we want from this citrate molecule at this point? What do you think we want to harvest from this citrate molecule eventually? What do you think? Think about it. What do we want from citrate? That's right, Wicket. We want hydrogens. Remember, there are nice, yummy hydrogens for us to collect and hand to the electron carriers. So let's see how that works. Citrate is going to be converted to isocitrate. Next, a very important step happens. Isocitrate is going to get oxidized, which means we're going to take some of its electrons away in the form of hydrogens. So we're going to give those hydrogens to NAD+, reducing NAD+ to NADH. This results in one of these carbons. You see this carbon right here and those 2 oxygens? Well, those leave as CO2. So one of the 2 carbons that entered the cycle has just left as CO2. So there's only 1 carbon left, right? Because look, 2 carbons entered, 1 carbon left, right? And once you form this NADH, where does the NADH go? Where does this NADH go once it's formed? That's right, Wicket. It's going to book it over to the electron transport chain. That's anytime NADH is formed, it just makes its way over to the electron transport chain to drop off that H, to drop off those electrons, to power the electron transport chain, and return in the form of NAD+ to do it again. It just shuttles back and forth, right? So now, after we've oxidized isocitrate, and isocitrate has lost one of its carbons as carbon dioxide, isocitrate is now converted to alpha-ketoglutarate. Alpha-ketoglutarate is this molecule right here. And look, what do we want from this molecule right here? Don't let Wicket beat you. What do we want from this molecule right here, do you think? That's right, Wicket. We want hydrogens. So we're going to take more hydrogens and we're going to hand it to NAD+, reducing it to NADH. This is going to cause this carbon and those 2 oxygens to leave as CO2. So at this point, how many carbons entered on acetyl-CoA? How many carbons entered? 2. And at this point, how many carbons have left? 2. So we shouldn't be losing any more carbons. We have officially run out of carbons on our glucose molecule. Okay, very interesting stuff. So let's take a look here and see what happened. Our alpha-ketoglutarate was oxidized to succinyl-CoA. The reason it has a CoA, by the way, obviously you don't need to know a lot of this stuff, but another coenzyme A has been added to alpha-ketoglutarate, and so what's left is succinyl-CoA. And now what's really interesting is this dance that occurs where a phosphate group, an inorganic phosphate, is transferred to succinyl-CoA, and then that phosphate group is transferred to GDP, phosphorylating GDP to GTP, and then GTP phosphorylates ADP to make ATP. So all that to be said, we just made an ATP molecule via substrate level phosphorylation. We moved a phosphate group around from organic molecule to organic molecule, and that's why this is known as substrate level phosphorylation. And we just made an ATP. And in the process, our succinyl-CoA is now known as succinate because it loses that coenzyme A, and succinate is then further oxidized. We take more of its hydrogens away, but this time we give it to a different carrier, right? We give 2 hydrogens to FAD. FAD picks up 2 hydrogens to form FADH2. And where do you think FADH2 goes with that cargo, that hydrogen cargo? It goes to the electron transport chain. Exactly. Succinate becomes oxidized to fumarate. Fumarate gets modified to malate. And what do we want? Finally, we want to pick the final carcass clean of hydrogens. So we're going to oxidize malate. We're going to give those electrons to NAD+, reducing NAD+ to NADH. Where do you think NADH goes? That's right, it goes over to the electron transport chain to drop off that H, and malate is now oxidized to oxaloacetate, which can start the cycle again. Now, I know that was a mouthful, but what does it all mean? You don't need to memorize each and every little step unless you're studying for the MCATs or you're in some kind of advanced biochemistry. But for a general biology course, you don't need to know every little baby step here. I don't expect you to know that, but I like to explain it so you can kind of visualize and see what's going on. Remember, 2 carbons entered, 2 carbons entered, 2 carbons left. A bunch of hydrogens were stolen and given to electron carriers, these NADH and FADH2, which all go to the electron transport chain. Remember, NADH visits complex 1 of the electron transport chain. FADH2 visits complex 2 of the electron transport chain, dropping off that cargo and then coming back for more. ATP was formed by substrate level phosphorylation, and remember, oxaloacetate was reformed at the end. Now, let me ask you this. This is only for one turn. This is only for one turn of the citric acid cycle. But what if I asked you, for every glucose molecule, what are the products of the citric acid cycle? Well, remember, it's everything you see here times 2. Because you had 2 acetyl-CoA, not 1. So if you have 2 acetyl-CoA, you actually get 2 rounds of the citric acid cycle, which means you form 1, 2, 3 times 2, 6 NADH, 1 times 2, 2 FADH2, 2 ATP, and how many CO2s? How many CO2s? 2 times 2, so 4 CO2s. Okay, and remember, what needs to be replenished at the end? Oxaloacetate. So let's go back to our overview sheet real quick and see if everything checks out. Look at our overview sheet. Again, I love this overview sheet. Look, 2 acetyl-CoA go into the citric acid cycle. And how many NADH are produced? 6. How many FADH2 are produced? 2. And where do they go? They go to the electron transport chain to drop off the electrons. How many CO2 are formed? 4. How many ATP are made? 2 by substrate level phosphorylation. And what needs to be replenished? Oxaloacetate must be replenished here at the end of the cycle. You see how beautiful that is? At this point, your glucose, your original glucose molecule, which was 6 carbons long, has fully been oxidized to CO2. Those 6 carbons of glucose, 2 of them left the system here as CO2, and the other 4 left the system here as CO2 as well. Okay, and all of the hydrogens from the glucose, you can think of the hydrogens. Look, you see this H here on these 2 NADH. You see these H's here on these 2 NADH. You see these H's and these H's here. Those were the H's from glucose. Does that make sense? The H's went to the carriers, to the electron transport chain. The C's and the O's left as CO2. Isn't that beautiful? So you have fully oxidized glucose, Its hydrogens were transferred to electron carriers and taken to the electron transport chain and ultimately ended up on water. Remember at the end of the oxidative phosphorylation? Oxidative phosphorylation include the electron transport chain and chemiosmosis. Remember NADH and FADH2 from the previous steps. That's what powers the electron transport chain to make the 28 ATP via oxidative phosphorylation. What was the terminal electron acceptor? Remember, the terminal electron acceptor was oxygen, which became reduced to water. You see, oxygen picks up the electrons at the end of the ride to produce water. So this is a beautiful process by which electrons began on glucose. They were transferred to the carriers, NADH and FADH2, which transferred those electrons to the electron transport chain, so they would flow from complex 1 or 2 through to complex 4. They were then picked up by oxygen, and those electrons ended up on water. The electrons started on glucose, and they ended on water. Isn't that neat? All right, did you get all that? We're back. So now you know that your 2-acetyl-CoA entered the citric acid cycle, giving you 2 rounds of the citric acid cycle, right? Not just 1 round of the citric acid cycle, 2 rounds of the citric acid cycle. And remember, during those 2 rounds, you formed 6 NADH, 2 FADH2, which, by the way, what do these do? Remember, the 6 NADH go to complex 1 to drop off those H's. Where do the 2 FADH2 go? They go to complex 2 to drop off the H's, to drop off the electrons. And what happens to the CO2? The 4 CO2s? You breathe them out, and you made some, couple of ATP here, so that's great, I guess, right? So that's there. Now, have we fully oxidized our glucose? Yes, at this point, we have fully oxidized glucose. Look at this. We started with C6H12O6. During glycolysis, we stole some of those H's. During pyruvate oxidation, we stole a couple more H's, and 2 of our 6 carbons left. 2 of our 6 carbons left as CO2 during oxidation of pyruvate, and the other 4 carbons left during the citric acid cycle. So think about it. We started with 6 carbons on glucose, and all 6 of those carbons leave as CO2 from our breath. That's what happens. We have by the end of the citric acid cycle, we have fully oxidized glucose to CO2, and we have formed 6 total CO2, which makes sense because we started with 6 carbons on glucose. So real quick, just to review, glucose is a high energy molecule, so is fat, and that's because it has all these yummy hydrogens. These hydrogens contain the electrons necessary and the protons to power the electron transport chain, so we need to take these H's. During glycolysis, we take some of those H's to make 2 NADH, which makes its way to the electron transport chain, feeding those electrons into the electron transport chain. During oxidation of pyruvate, we take a couple more H's, make a couple more NADH, so that we can transport those H's to the electron transport chain. During citric acid cycle, we take a bunch of H's. We make 6 NADH, 2 FADH2, so that we can feed those into the electron transport chain. So do you see what's happening here? We have been oxidizing glucose. We oxidize glucose so that we can reduce NAD plus to NADH or FAD to FADH2. So we're transferring H's from glucose to the carriers, and then those carriers are transporting those H's to the electron transport chain, and then those electrons are being picked up by oxygen to form water. So did you see what happened? I just want to make this clear before we move on. I want to make this clear. Can we follow the journey of the electrons here? Remember when I said cellular respiration is just a series of redox reactions reduction, oxidation, reduction, oxidation. I just want to drive this home real quick for you. The electrons started on glucose, right? You see, I'm just going to talk about the electrons instead of the H's, okay? The electrons started on glucose or fat, okay? The electrons are handed to either NAD plus or FAD during one of these three processes. When those electrons are given to the carriers, they become the reduced form of the carriers, right? They have the H's, they have the electron cargo. Those electrons then go to the electron transport chain. NADH hands its electrons to complex 1. FADH2 gives its electrons to complex 2. Those electrons go from complex to complex, right? You see, those electrons hop from complex to complex, cofactor to cofactor. And who picks up the electrons at the end of the ride? That's right, the terminal electron acceptor. Oxygen picks up the electrons to form water. Did you see how this is the story of a traveling electron, right? The electrons begin on glucose. The electrons are transferred to the carriers. And by the way, the enzymes that transfer electrons to the carriers are called dehydrogenase enzymes, in case you're curious. So the electrons started on glucose. The electrons were transferred to the electron carriers, either NAD plus or FAD. They were reduced to NADH or FADH2. The electrons visit complex 1 or complex 2. The electrons flow from complex to complex. The electrons are then picked up by oxygen to form water. So isn't that neat? The electrons went from glucose to carrier to electron transport chain to oxygen to make water. That was the full traveling schematic there. That was the journey of the electron, right? So you can think of cellular respiration as kind of the story of a journey of electrons, right? And because of this journey of electrons, this powers this powerful pump system to pump protons. Those protons then chemiosmose, exerting a proton motive force, spinning ATP synthase to make the lion's share 28 ATP. 28 ATP. So in this next little clip, I'm going to explain to you why 28 plus 2 plus 2 gives you 30 net ATP in eukaryotes, whereas 32 net ATP in prokaryotes. I'm going to explain that next. So to answer why eukaryotes net only 30 ATP while prokaryotes net 32 ATP, it all comes down to this step right here. Both eukaryotes and prokaryotes make 2 ATP here during glycolysis via substrate level phosphorylation. They make 2 ATP here during citric acid cycle with substrate level phosphorylation, and they make 28 ATP here at oxidative phosphorylation, during the final step here with ATP synthase, 28. So 28 plus 2 is 32 ATP. So prokaryotes get to keep all 32 ATP. However, in eukaryotes, you see this step here. You see how pyruvate needs to enter the mitochondrion? Well, that is a type of active transport, and it requires 2 ATP to be used, right? So to get the 2-pyruvate into the mitochondrion, we need to invest 2 ATP. So if we're making 32, but we have to use 2 to get pyruvate into the mitochondrion, then in essence, we didn't really net 32, we netted 30. And do prokaryotes have a mitochondrion? No, remember, prokaryotes don't have mitochondrion. So prokaryotes, they get to keep these two, and these 28. They don't need to spend ATP to get pyruvate into the mitochondrion because they don't have a mitochondrion. All these steps occur in the cytoplasm. I hope that makes sense. So for the last part of this lecture, I want to tell you about these concepts, the important concepts of the difference between aerobic respiration, also known as aerobic cellular respiration, anaerobic respiration or anaerobic cellular respiration, and fermentation. One thing I want you to know up front is that all of these are separate concepts, right? Anaerobic respiration is not aerobic respiration. Fermentation is not anaerobic respiration. Each one is an individually different concept. And I just want to drive home, first of all, let's just explain the difference between aerobic respiration and anaerobic respiration. Okay, so what I've been explaining this whole time of glycolysis, oxidation of pyruvate, citric acid cycle, and oxidative phosphorylation, these 3.5 steps that I've spent this time explaining, where I said that oxygen is the terminal electron acceptor of the complex 4 of the electron transport chain. This is known as aerobic respiration. Aerobic respiration, oxygen is your TEA. It's your terminal electron acceptor. It's the one that picks up the electrons at the end of the ride, okay? And that's what aerobic respiration means. By the way, what does respiration mean? When you hear the term respiration or cellular respiration, I want you to know this. Anytime you see a term called respiration, like aerobic respiration or anaerobic respiration, when you see the word spelled out respiration, what you need to know is that this involves glycolysis plus oxidation of pyruvate plus citric acid cycle plus the oxidative phosphorylation, which includes all of this stuff, electron transport chain, ATP synthase. Everything you see here is respiration, right? So aerobic respiration means everything you see here and your terminal electron acceptor is oxygen. But what's anaerobic respiration then? What's anaerobic respiration? This is where my students can kind of get confused at times. They'll say, anaerobic respiration is without oxygen, because anaerobic means without oxygen. But that's okay, but that's only a fraction of the definition, the fraction of the explanation. So what is anaerobic respiration? That means that this is happening, but without oxygen, right? Without oxygen. But didn't I tell you something? Okay, think about this real quick. Didn't I tell you that if there's no oxygen, then this shuts down? And that's true. This whole thing shuts down. If this shuts down, is that still respiration? Because remember, what did I tell you respiration is? Respiration means glycolysis is working, oxidation of pyruvate is working, citric acid cycle is working, oxidative phosphorylation is working. These are all working. So if I just took away oxygen, that wouldn't be respiration, would it? This would shut down, and everything except glycolysis would shut down, by the way. Did you know that? If I run out of terminal electron acceptor, everything shuts down except glycolysis. We'll get to that. But what's anaerobic respiration? How can I do all of this? Let me ask Wicket. Let me ask you to think about this for a second. I just want you to think for a second. And I'm going to ask Wicket to answer too. Hopefully you can beat him to the punch. How can I do all of this without oxygen? How can I do all of this without oxygen? If I just took oxygen away, well, then it would shut down, right? But how would I do all of this without oxygen? What did you say, Wicket? Wicket is a genius, you guys. Wicket is saying that what if something else besides oxygen served as our terminal electron acceptor, such as reduced sulfate, SO4 2-, right? So, a molecule other than oxygen serves as the terminal electron acceptor. then if this other molecule were to pick up the electrons, well, would that work? Would that work? Okay, think about that. If I used a different electronegative molecule to pick up the electrons at the end of the ride, would the ride shut down? No, the ride would keep going, right? So that's what I want you to know. That's what I want you to understand. In anaerobic respiration, glycolysis still works, oxidation of pyruvate works, citric acid cycle works, and oxidative phosphorylation works. But the only difference is what? The only difference is that instead of oxygen being our terminal electron acceptor that picks up the electrons at the end of the ride, some other molecule that's electronegative, such as reduced sulfate, is our terminal electron acceptor. Does that make sense? So it's a molecule other than oxygen that serves as the terminal electron acceptor. Now, let me ask you this. Do you think that humans like you and me, are capable of anaerobic respiration? That's right, Wicket. We are not. But let me tell you this, we aren't the only creatures on the planet. There are some prokaryotes, bacteria, archaea, and did you know there are some creatures that don't breathe oxygen at all? They breathe something like sulfate, right? So, you know, if you've ever seen those sulfur springs where it's like hot and bubbly sulfur springs, well, there's creatures living in there and they're breathing not oxygen, they're breathing sulfur. So they're breathing something like sulfate. So humans, what you should know is that humans are not capable of anaerobic respiration. It's impossible. That would mean that we breathe something other than oxygen. No, we are aerobic respirators. Does that make sense? We breathe oxygen, okay? We can't switch to breathing sulfate if oxygen becomes unavailable, right? But there are some creatures that breathe sulfate or nitrate, right? Or something else, right? So these creatures breathe something other than oxygen. They use something other than oxygen as their terminal electron acceptor. So the electron transport chain never shuts down. Is that neat? So you should know that the only difference between aerobic and anaerobic respiration that I need you to know about is that the terminal electron acceptor is different, right? It's a different molecule. And humans are not capable of anaerobic respiration at all. All right, so you might be wondering, well, Dr. D, what in the world is fermentation then? All right, so I'm going to explain fermentation. Fermentation, I want you to know this because students mess this up all the time, okay, you guys? Fermentation is not anaerobic respiration. You hear me? Fermentation is not anaerobic respiration. They are not the same thing. Remember, anaerobic respiration means everything here is working great, except your terminal electron acceptor is something other than oxygen, okay? That's not what's going on in fermentation. Let me explain fermentation, okay? Look at this. What was our terminal electron acceptor for us humans and cats, Wicket? Okay, oxygen, right? Oxygen. And what did oxygen become reduced to? To water, right? Okay, during this process. Now, let's say you and I, we, decide to run a marathon. And, I'm, let's say a couch potato. I'm not usually running at all that much, right? So I'm not training for this marathon. But let's say I want to go run a marathon. The first thing that's going to happen after a couple miles is I'm going to just start breathing really, really hard. You know why? Because suddenly I'm exercising and my body is trying to produce a bunch of ATP to keep me going, okay? To keep my body going, I need to produce more ATP. This means that I'm going to burn more sugars and fats, but what do I need? Why am I breathing so hard? I'm breathing so hard because I need more oxygen to keep this higher rate of cellular respiration going. You see what I'm saying? So if I'm exercising at the gym too hard, or if I'm trying to run a marathon, I'm going to start panting. Oh my gosh, panting, because I need more oxygen to power this process, and I need to breathe out more CO2 to get rid of that CO2, and I need to breathe in more oxygen, breathe out more CO2, because I'm trying to make more ATP. You see the problem here? So eventually, what's going to happen? Well, I'm breathing as hard as I can, but I may not be getting as much oxygen as I need, right? I may not getting as much oxygen in my blood as I need to keep this going, right? Because this is happening in each and every one of my cells. Each of my cells is doing this cellular respiration, so I need to breathe in oxygen and get it to all my cells in my body. I just can't do that if I'm not training enough. So what happens? Remember, if there's not enough terminal electron acceptor, do these electrons get picked up at the end of the ride? That's right, Wicket, they don't. And that means that the electrons pile up like a freeway traffic jam. And when the traffic jam's all trafficked up, right, what happens? This whole thing shuts down, right? Oxidative phosphorylation shuts down. What else shuts down? Remember I mentioned it earlier. What else shuts down? That's right, the citric acid cycle shuts down. What else shuts down? Come on, what else shuts down? Oxidation of pyruvate shuts down. Look at that. And what's the only thing that keeps chugging along? Glycolysis, right? Remember, glycolysis occurs in the cytoplasm. Okay, only glycolysis is chugging along. So when I'm doing too much activity, when I'm doing too much exercise, okay, I'm doing too much exercise, I'm not getting enough oxygen. Everything shuts down in my body, in my tissues, in my muscles. Everything shuts down except what? Except glycolysis. Okay? So in this case, instead of making a nice profit, instead of making 30 net ATP per glucose, how many ATP am I making per glucose during fermentation? By the way, that's what fermentation is. Fermentation is when I don't have enough oxygen, I don't have enough terminal electron acceptor, and everything shuts down except glycolysis. That's what the definition of fermentation, right? Fermentation. Only glycolysis is going on because of lack of terminal electron acceptor, right? So instead of 30 net ATP, how many ATP am I making net during fermentation? 2, right? 2 net ATP. So I'm making 2 ATP, I'm making 2 NADH, I'm making 2 pyruvate, because that's all glycolysis is. So that's what's happening during fermentation. During fermentation, I'm not getting enough terminal electron acceptor, everything's shutting down except glycolysis. So I've gone from making a whopping 30 ATP per glucose molecule to only 2 ATP net per glucose molecule. That's not good. Can your body withstand that for a long period of time? Like if you were being paid $30 an hour, can you now survive on only $2 an hour, right? You're gonna come up short. Does that make sense? And that's why if I tried to go run a marathon right now and I haven't trained, I'm gonna start panting and panting and panting and breathing hard, trying to get more oxygen. But eventually, this is what's gonna happen. I'm going to switch to fermentation, and I'm only making 2 ATP where I was making only, I was making 30 before, now I'm only making 2, and I can't withstand this. Things are going to start hurting and cramping up and spasming, and I'm going to fall over. I can't finish that marathon. You see what happens? So this is fermentation. You see how fermentation is not anaerobic respiration? In anaerobic respiration, the identity of my terminal electron acceptor is something else, and humans aren't capable of anaerobic respiration. But humans are capable of fermentation, because if we hit the gym too hard or go for a run too hard, we will run out of oxygen in our muscles or run short, and our muscles are going to switch to, just doing glycolysis, doing fermentation. So in the next little clip, I'm going to explain exactly how fermentation works. By the way, before I explain fermentation in a little more detail for you, I'm going to explain why students confuse fermentation with anaerobic respiration all the time. And that's because it's tricky. The terminology is tricky. Sometimes fermentation is known as a anaerobic process, okay? So it's true that fermentation is anaerobic. It's true that fermentation is an anaerobic process. But let me ask you this. Is fermentation anaerobic respiration? Right? Think about it. Respiration means that all of those steps were happening. Remember, glycolysis, oxidation of pyruvate, citric acid cycle, oxidative phosphorylation, right? All this stuff's happening. All this stuff's happening. That's anaerobic respiration. That's not what's happening during fermentation. The reason why fermentation is called an anaerobic process is because anaerobic just means without oxygen. And yes, fermentation is a process that happens when there's no oxygen. So yes, technically, fermentation is an anaerobic process, but it is incorrect to call fermentation anaerobic respiration because the respiration part of that term means all those steps are working, which obviously are not working. So I hope that makes sense. It's okay to call fermentation an anaerobic process, but it is not okay to call fermentation anaerobic respiration. Just wrong, right? So now let me explain fermentation in a little more detail. And the type of fermentation I'm going to explain on the board right now is the exact type of fermentation that happens in your body. You know, when you hit the gym too hard or you run that marathon when you're not trained, right? Remember what happened when I started running really hard and I was not prepared for the marathon, right? Remember, breathing hard, not getting enough oxygen to my muscles. What happens? Remember, everything shuts down except what? Except glycolysis, right? And do you remember how glycolysis works? During glycolysis, glucose is broken down into what? Remember 3 products of glucose. Remember this? Glucose is broken down into 3 carbon pyruvates, 2 molecules of pyruvate. How many net ATP are formed during fermentation? 2. And what else is formed during glycolysis, right? During glycolysis, you have 2 pyruvate, 2 ATP, and how many NADH? 2. Remember, that's just glycolysis. We learned glycolysis earlier, right? During glycolysis, you make 2 pyruvates, 2 ATP, 2 NADH. Fair enough? Okay. But now, did you know that, during fermentation, when I hit the gym too hard, not only does stuff start cramping up, but if you've ever been in sports and if you've ever had a coach, your coach might notice that you're spasming and cramping up, and the coach might ask you to sit down, okay? And the reason the coach sits you down is because they explain to you that you might have what's called lactic acid buildup, right? During too much exercise, my muscles can build up lactic acid. Some of you may have heard of this in the past. Well, you know why? Because lactic acid is a fermentation product in animals, right? Here's how it works, okay? Let me explain why lactic acid is built up. Because is lactic acid a product of glycolysis? No. Glycolysis results in 2 pyruvate, 2 ATP, and 2 NADH, right? So why are we producing lactic acid if all that's happening is glycolysis and lactic acid is not a product of glycolysis? Well, let me explain, okay? Here's how it works. You ready? Okay, here's how it works. Look, normally I have plenty of sugar to keep this process going. I have plenty of sugar, okay? And I keep converting the sugar to pyruvate, and I keep making 2 ATP. That's great. But here's what I need you to understand, okay? My NAD+, the NAD+, this oxidized carrier, the oxidized form of the carrier, NAD+, is required. It's required for glycolysis to continue, right? You know why? Because NAD+ needs to come along, and during glycolysis, it picks up 2 electrons and a proton to become NADH, right? Do you remember that? NADH? So I need NAD+ because it gets reduced to NADH during glycolysis. Do you follow me so far? But here's what I need you to understand. Your pool of NAD plus is not very large. Your cells don't have an overwhelming amount of NAD plus at any time. Your mitochondria are not stuffed with NAD plus. This is a limited resource, okay? And here's the thing. When that resource becomes reduced to NADH, where does the NADH go at this point normally? Let me ask you this. Where does NADH normally go? Well, where NADH always goes, right? It goes to the electron transport chain. Which complex does NADH visit at the electron transport chain? Complex 1 or 2? Yeah, complex 1. Complex 1 of the electron transport chain. And then it drops off that H, right? It drops off those 2 electrons and the proton. And what does it become? What does it become? Doesn't it become oxidized back to NAD+? And doesn't NAD+ shuttle back to pick up more H's? You see how it normally works? You guys follow me? Normally, during respiration, NAD+ is reduced to NADH by glycolysis. NADH goes to the ETC, the electron transport chain complex 1. It drops off its H, it drops off its 2 electrons and a proton, gets oxidized back to NAD+. NAD+ can come back and do it again. That's how we normally regenerate our pool of NAD+, so that the cell does not run out of NAD+, so the mitochondria doesn't run out of NAD+. Does that make sense? You guys follow me so far? That's just a review, right? But let me ask you this. During fermentation, I've hit the gym too hard. During fermentation, is the ETC working? Is the electron transport chain functional during fermentation? That's right, Wicket. It is not functional, right? So there is no ETC, okay? So is there a place for NADH to go drop off its H? No, right? So think about this for a minute. I just want you to think about this. I told you that there isn't a ton of NAD+, but you need NAD+ to keep doing glycolysis. But what's happening during fermentation is that your limited NAD+, all of it's coming along and picking up electrons and becoming NADH. So all of your NAD+ is becoming reduced to NADH. And the NADH don't have any place to give the H, right? To replenish what? We need a way of replenishing. We want to replenish NAD+. Does that make sense? If we run out of NAD+, we are so, out of luck, because then glycolysis cannot continue. Does that make sense? So we need a place to dump these H's, because we can't take it to the electron transport chain. So you know what we do? We give the H to pyruvate. Look, give the H to pyruvate. Give the H to pyruvate. And what does it become? When you give pyruvate those H's, those pyruvate become 2 lactic acids, also known as lactates. Lactate. Isn't that cool? Did you see what just happened? Did you see? Okay. We needed a place to dump these H's so we don't run out of NAD+, because we want more NAD+, okay? So we gave those H's directly to pyruvate, and pyruvate got reduced. Pyruvate got reduced to lactic acid. And this is why during strenuous exercise, your body will start building up lactic acid. Why? Because your body did not want to run out of the limited resource NAD+. And the only way to do that during fermentation is to reduce pyruvate to lactic acid, thereby replenishing NAD+. Does that make sense? So during lactic acid fermentation, your body is only doing glycolysis. You're only producing 2 net ATP. And the limited resource, the limited factor that needs to be replenished is NAD+. And how do you do it? By using NADH to reduce pyruvate to lactic acid. Because when you give those H's to pyruvate, pyruvate becomes lactic acid. Okay, y'all ready for the final concept of this chapter? Alcohol fermentation. Did you know that not all creatures do the exact same fermentation? So you and I, as humans, we undergo lactic acid fermentation, which I just explained. When you hit the gym too hard, not enough oxygen, you produce lactic acid as a fermentation product. But different creatures on the planet Earth, they can produce different products from fermentation. So for example, there's a myriad of different ones, but I'll give you a common example called alcohol fermentation. Some creatures like yeast, for example, Saccharomyces cerevisiae, which is baker's yeast. Yeast, when it has oxygen, it does aerobic respiration, right? So if I gave yeast oxygen, plenty of oxygen and glucose, the yeast would undergo respiration. But what would happen if I shut the air off or put the yeast in a closed bottle, right? Then the yeast would switch to what? If I were to block off the oxygen, could the yeast continue to do aerobic respiration, which includes glycolysis, oxidation of pyruvate, citric acid cycle, and oxidative phosphorylation? No, because I've closed the bottle, right? I've put the yeast in the bottle and I've closed the bottle. Now the yeast don't have any oxygen, and so they run out of oxygen. And what do they switch to? The yeast will switch to fermentation, right? But they do a different kind of fermentation. They do alcohol fermentation. So I'm just going to explain that real quick, and that'll be the end of the chapter. So again, during fermentation, now this time it's alcohol fermentation. Again, there's not enough oxygen. The yeast do not have enough oxygen, so they switch to just glycolysis. They switch from aerobic respiration to fermentation, so they're only doing glycolysis. That means they're converting a glucose into 2 pyruvates. They net 2 ATP, and remember, they're converting 2 NAD pluses to 2 NADHs, right? This is very similar to what we just discussed about lactic acid fermentation. And remember, I want to ask you this. What's the limiting factor during fermentation that needs to be replenished? Can you beat Wicket? That's right, Wicket. It's NAD+. Remember, NAD+ needs to be replenished. If you don't replenish NAD+, you're out of luck, because all of it gets converted to NADH. You run out of NAD+, and then, you know, you're not even making 2 ATP for every glucose, which means that's like pretty likely death, right? So how do you combat that? Remember, you and I as animals, we solve that problem by giving the H to pyruvate directly, and pyruvate becomes lactic acid. Remember that? But the only difference with alcohol fermentation is one extra step. First, the pyruvate is converted to acetaldehyde. To acetaldehyde. What yeast do, for whatever reason, yeast cleave their pyruvate, releasing CO2. They release CO2. They first convert their pyruvate to acetaldehyde. Then they give it the H from NADH, right? So they give it that H. That means that NADH gets oxidized back to NAD+. And if you give acetaldehyde an H, it becomes what? It becomes 2-ethanol. It becomes drinking alcohol. It becomes alcohol, and that's the same as drinking alcohol. Isn't that neat? So It's the same basic principle as lactic acid fermentation, but what's the only difference? You still only make 2 ATP. You're still only doing glycolysis. You still only make 2 ATP. You still need to replenish the NAD+. The only difference is in you and me in lactic acid fermentation, pyruvate is directly reduced to lactic acid, whereas in alcohol fermentation, pyruvate is first converted to acetaldehyde, and then that is reduced. Acetaldehyde is reduced to ethanol, right? So isn't that neat? And this is how alcohol is brewed. Did you know? Like, how do you make wine? How do you make wine? Don't you take grape juice? You could add some sugar if you want, so you got grape juice, which has natural sugars, or you could add a little more sugar, so you've got sugar, okay? And then you got to add your yeast, right? Your yeast cells, which is Saccharomyces cerevisiae. And then what do you need to do with that container? Can you just leave that container open if you want to make wine? Or do you have to close the barrel, like plug the barrel up? Remember, yes, you have to plug the barrel up. Why do you need to plug that barrel up if you want to make wine? Well, because you want the yeast to switch from aerobic respiration, where they're not going to be making any ethanol, to fermentation, where they are going to be making ethanol. Why? Because during fermentation, those yeast cells are just doing glycolysis. So the yeast are taking the glucose from the grape juice, or from the sugar that you put in the grape juice. They're converting that glucose in the grape juice to pyruvate. They're getting 2 ATP. They're converting NAD+ to NADH. They need a way of getting their NAD+ back. So what do? Instead of giving that H directly to pyruvate like we do, they first convert the pyruvate to acetaldehyde, and then they reduce the acetaldehyde to ethanol, thereby making wine, thereby making the alcohol in wine. Isn't that neat? And yes, those barrels can get bubbly, right? Because there's CO2 produced as well. Isn't that neat? So CO2 is produced during this process. So now, tell me you're not impressed. Tell me it's not interesting, right? Because students, a lot of times cellular respiration can be dull, bland, boring, what have you. Yeah, it can be. But at the same time, look what you've just learned. You've learned why you breathe oxygen. You've learned why you breathe out CO2. You've learned how you gain weight or lose weight. You lose most of your weight through exhaling CO2. You've learned why you diet, because you don't want to replace those sugars and those fats immediately after you've converted them to CO2. You've learned why when you hit the gym too hard, you get cramps and everything gets all lactic acidy, right? Because you will need to replenish your NAD+ by reducing pyruvate to lactic acid. And you've learned the principles behind alcohol fermentation. Yeast cells, they're doing fermentation of their own. They need to replenish NAD+. So they first convert their pyruvate to acetaldehyde and then reduce that to ethanol. Now, I think that's a pretty interesting chapter.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.